Hello, everyone. Thank you for joining the National Stock Assessment Science Seminar. My name is Bailey, and I coordinate this monthly seminar series that highlights interesting advances and topics in stock assessment science. Today, we are pleased to have John Swenson with us. John is a late-stage PhD candidate in the Molecular Ecology and Conservation Lab at the University of Massachusetts. His work focuses on informing the conservation and management of vulnerable marine species using the closed-kin mark-recapture framework. In today's seminar, John will discuss how to design and implement a successful CKMR project to estimate abundance and mortality of data-limited marine species. Without further ado, I'll pass it over to John.
So thank you all for coming. As I said, my name is John, and I'm a late-stage PhD candidate at the University of Massachusetts. And at the core of my dissertation is a method called close-kin mark recapture, which was introduced to the world in 2016 by Mark Brovington, Hans Skog, and Eric Anderson, and has been growing in popularity ever since. Close-kin mark recapture is a genetic-based method for estimating population abundance and mortality that is not subject to many of the limitations that challenge more conventional methods like capture mark recapture or CPUE. Since abundance and mortality are two of the most important parameters for stock and species assessments, close-kin mark recapture has the potential to enable or boost assessments and monitoring programs in situations that were previously infeasible or challenging. So the method is quite exciting, but it is also quite complicated, and there is a lot of potential for things to go wrong. But as long as we're aware of and account for potential hurdles during our project design, then close-kin mark recapture can be a very powerful tool. When I think of close-kin mark recapture, also known as CKMR, I think about it as a method that sits at the intersection of this Venn diagram. CKMR combines cutting-edge genomics technologies with some of the latest advances in computational biology and statistics. And whenever I hear about a talk related to CKMR, I do my best to attend, and I always enjoy them, and I always learn something new. But most of the talks that I've seen so far have come from people whose backgrounds are primarily in population modeling, and so that third of this Venn diagram has tended to be the emphasis. And that's important and appropriate because there are many things that need to be considered to construct and apply a robust CKMR model. But I bring a slightly different perspective to the table. My background is primarily in genomics and bioinformatics, the other two thirds of that Venn diagram. Before I started my PhD, I did a research-based master's degree where my advisor, Karen Crow, and I used RNA sequencing to look into the genetic underpinnings of the manta ray body plan. We also compiled the first ever developmental staging scheme for the myeliobatid family of rays. These are images of little developing cow-nose ray embryos, and their morphological and genetic development is fascinating. If you're interested, the associated paper is open access. But the reason I bring that up now is to highlight that my master's degree introduced me to the universe of genomics and bioinformatics, and it gave me the opportunity to start building up those skill sets. And then for my PhD over the last 6 years, I've been building on those skills and studying ways to integrate genomics outputs into population models that can help inform conservation management. And the CKMR framework is where I found that integration. So one of my overarching goals has been to understand and build my capacity to implement every step of a CKMR project, from sampling design through kinship identification to model fitting and validation. And my research so far has focused on elasmobranchs, a group for which genomics analyses can be fraught. Sharks and rays tend to have large genomes, slow mutation rates, and lots of repetitive elements while also lacking in genomics resources. What this means is that the genetic analyses that are prerequisites to fitting a CKMR model can be quite challenging. And so given my experience and background, this is one of the main topics that I want to talk about today. Things that we need to consider upstream of model fitting that can make or break a CKMR experiment. So to give a roadmap for what the next 45 minutes will look like, I'll start with some background so that we're all on the same page about what CKMR is and what it can and cannot do. And I'll also give some baseline information about genotyping as well, so we all have shared terminology going forward. Then I'll share some examples of real-life CKMR applications, focusing on 3 that I think are particularly creative and demonstrate the potential of CKMR to do even more than estimate abundance and mortality. And then after this, we'll step back and talk about considerations for project design, ending with a flowchart of the major steps involved in a large-scale CKMR project. And then finally, I'll highlight some ongoing applications that I'm involved with and then connect these back to our project design flowchart to show where they land in the process. So we are going to cover a lot of ground. So without delay and with this roadmap in mind, let's go. So I'd like to start by defining CKMR, and I'm going to do this in layers over the next 10 minutes or so, appealing to different learning styles. So if anybody feels a bit lost after this slide, stick with me. I hope it will make sense about 10 minutes from now. So CKMR is a genetics-based method for estimating absolute adult abundance, mortality, and other population parameters. Fundamentally, it's a modified version of capture, mark, recapture. But rather than relying on recaptures of individuals, CKMR relies on probabilities of kinship or relatedness. And these kinship probabilities are based on the reproductive output of individuals relative to the reproductive output of the population. So for every individual we sample, say individual A, we loop through all of the other individuals that we sampled and ask what is the probability that individual B is related to individual A, given relevant covariates like age and sex. The fundamental idea behind using genetics to assign relatedness comes from a concept called identity by descent, which means that offspring share a predictable proportion of their genome with their parents. This means that we can think about the DNA of offspring as genetic tags of their parents. So if we capture and identify an individual's offspring, then that is an instance where we have captured the parent's genetic tag. If we capture and identify a second offspring from the same parent, then we have recaptured that parent's genetic tag. If we were to capture a parent directly and then capture one of its offspring, then that is another way that we can capture and recapture the parent's genetic tag. CKMR is a highly flexible framework, and it can technically make use of any type of relationship, contingent on the ability to reliably identify that relationship and construct an appropriate kinship probability. The more distant the relationship, the harder these 2 criteria are to meet. So while a CKMR model could technically accommodate cousins, most practical applications have focused on first or second order relatives like parent-offspring pairs or half-siblings. In addition, CKMR can generate a time series of abundance estimates from a single sampling occasion. This is because in CKMR, abundance estimates are backdated to the birth years of the sampled individuals. This is a key point that will come up several times. The most important unit of time in CKMR is the birth years that are represented in our samples. So when we think about CKMR and the relevant time units, we should think in terms of birth years more so than capture years. CKMR is a very adaptable and flexible framework, and the outcomes that we can expect depend largely on project design and model construction, but there are a few steps that are generally a part of any CKMR project. A typical CKMR experiment will involve collecting tissue samples, preferably across a population's range, as well as some metadata. After we've collected tissue samples, then we extract DNA from each of the samples and put it through a battery of lab work, sequencing, and bioinformatics analyses to ultimately determine who's related to whom and how. And then we construct a pairwise comparison matrix where we compare every sample to every other and assign a kinship probability to each comparison based on covariates like age and sex. As I mentioned earlier, pretty much any relationship could technically be used in a CKMR model, but the 2 types that have been most commonly employed so far are parent-offspring pairs and half-sibling pairs. So I'm about to put up a couple of equations, and I did not define all of the terms. I'm just going to move through them quickly to highlight a couple of key points. But if you would like more details about the derivations of these equations, check out the Brovington, Skog, and Anderson paper that I highlighted on the first slide. So if we make various assumptions, such as reliable aging, constant fecundity, and no stock structure, then this equation describes the probability that any 2 randomly sampled individuals are related as maternal parent-offspring pairs if sampling is non-lethal. If sampling is lethal, then this equation becomes simpler. And this equation describes the probability that any two randomly sampled individuals are related as maternal half-siblings. In other words, they share a mother but not a father. And the reason I wanted to show these equations primarily is to emphasize the prevalence of the terms YI and YJ. YI and YJ refer to the birth years of the individuals in each comparison. And if we look at the abundance terms, we see that in each case, abundance is estimated in the birth year of the younger individual in each comparison. So knowing the birth year of individuals that we sample is important for CKMR, and it means that we need to be able to age sampled individuals with some degree of precision. Now, let's assume for demonstrative purposes that this basic half-sibling probability is appropriate for our system. In real applications, we will almost always need to modify these basic equations in one way or another. But once we define an appropriate kinship probability, we can apply it to every pairwise comparison among our samples. And then the number of observed kin pairs can be described by a binomial distribution, where the number of trials corresponds to the total number of pairwise comparisons among our samples, and the probability of success equals the kinship probability. There are, of course, other distributions that could be used. I've seen others use Poisson distributions, but I've had success with the binomial. And so this is a very high-level overview of the steps involved in a CKMR project, and we'll dig into more detail later. But for those who are more visual, I want to offer a little illustration to build intuition about how CKMR works. So here, let's imagine that we have adults up top and their offspring down on the bottom, and the dotted lines represent parentage. Let's imagine that we sample 4 juveniles from this population. If we trace parentage back through the solid black lines, then we can see that we have 2 instances where sampled individuals share one parent but not the other. In other words, we have 2 half-sibling pairs. In the CKMR framework, this means that we have essentially caught and then recaptured the adult's DNA. And that framing is what allows us to interpret patterns of kinship in a mark-recapture framing. So let's wipe the slate clean now and imagine that we employ a different sampling scheme where samples are more evenly split between adults and juveniles. In this case, we may sample 2 juveniles and 2 adults. If we again trace parentage back through the solid black lines, we see that there are 2 instances where we have directly captured the adult's DNA by capturing the adult, and then we've recaptured his DNA by capturing its offspring. So we have 2 parent-offspring pairs. In addition, eagle-eyed folks might have noticed that the 2 sampled juveniles are also related as half-siblings. So in the CKMR framework, this means that we have caught and recaptured the genotypes of these 3 adults. If the population was large, we would expect to find fewer relatives. In other words, we would have fewer recaptures of the adult DNA than if the population was small. That's how CKMR works intuitively. And we can absolutely combine different kinship probabilities to increase the power and potential of our model. So the method is exciting, and it has some major advantages relative to more traditional abundance estimation methods. A big one is that there is no need to recapture any individual animal with CKMR. This means that we can use CKMR in circumstances where samples are obtained lethally, like in fisheries, or where individual animals are unlikely to be observed more than once. There are also no issues with tag loss or tag reporting because the tags are encoded in DNA. And there is, in fact, no need to observe adults at all to gain insight into the adult population dynamics. This can be very helpful for species where habitat use shifts during ontogeny. Many sharks, for example, spend time near shore as juveniles, where they're readily available for sampling before they transition into a more pelagic and difficult to access environment as an adult. These are definitely not all of CKMR's strengths, but they are 3 of the big ones that can give CKMR a leg up over other abundance estimation methods. However, CKMR is, of course, not a panacea, and it also has its limitations. And one of the biggest fundamental limitations of CKMR is that it can only inform on the adult portion of the population because the framework is based on reproductive output and only mature individuals produce offspring. It can require considerable biological knowledge and technical expertise to apply properly. Biologically, we need to know enough about our system to set expectations for reproductive output. And technically, we need to have the skills to reliably genotype, assign kinship, and fit a potentially complicated population model. And CKMR does require age data, or at a minimum, a reliable age length curve. Fortunately, a study came out just a couple of months ago where Felix Pietersma and colleagues found that as long as we have a decent age length curve and account for that in our model, then CKMR can produce unbiased estimates of abundance, even if the age length curve is not very precise. In circumstances where we don't have a reliable age length curve, some colleagues and I ran simulations showing the bias can start to accrue if there is a lot of uncertainty in age assignment that's not accounted for in the model. And in this case, if we cannot just back calculate the birth year of sampled individuals, one option is to focus sampling on young of year if they're available, which are typically easier to age. And we found in our simulations that as few as 4 cohorts was sufficient to produce robust estimates of abundance, even for populations that breed bi or triennially. There are a few types of species for which CKMR is unlikely to be ever useful, like super high abundance species like krill, or super rare species that you're unlikely to ever observe, and long-lived, hard-to-age species that cannot be sampled while young. But in most cases, the limitations are likely to be related to sample availability, funding, and technical expertise. So to pivot a little bit, before we go further, I want to give a little bit of background information about genotyping so that we have a shared lexicon going forward. Most modern applications of genetics and genomics in evolution and ecology rely on something called SNPs, or single nucleotide polymorphisms. SNPs are genetic variants. In other words, they're a unit of variation in the genome, which means that for a given site in the genome, one individual animal might have an A and another might have a T. SNPs are ubiquitous across the genome. When I use the word genotyping is the process of calling SNPs. So for any given site in the genome that we know varies among individuals, we ask, is this individual an A, a T, a C, or a G? And since diploid organisms have 2 copies of DNA, individuals might also have 1 copy of DNA that's an A and one that's a T. The data requirements to generate individual genotypes like this tend to be greater than the data required to calculate allele frequencies and delineate stock structure. But because SNPs are ubiquitous across the genome, we can leverage the power of thousands of SNPs at once, and we're able to detect first and second degree relatives like parent-offspring pairs, full siblings, and half siblings. And if you have enough SNPs, you can identify relatives that are even more distantly related. Crucially, the more distant the relationship, the more statistical power we need to disentangle relatives from unrelated individuals. So, for example, we need more genetic markers, or more SNPs, to identify half-siblings than we do to identify parent-offspring pairs. And in general, the data requirements for relatedness tend to be greater than they are for stock structure analysis. And the lab work in bioinformatics required to generate individual genotypes and kinship using thousands of SNPs involves some rather complicated lab protocols, as well as detailed and specific bioinformatics work. So when planning a CKMR project, I would not necessarily assume that any genetics lab can do the required genetics work. Many can, but there should be some detailed conversations beforehand, at least, to ensure that any lab that's recruited has the specific expertise required. So returning to our roadmap now, I'd like to step back out of the theoretical and jump into the practical, and share some examples of real-life applications of CKMR that I've found particularly ingenious. And if you follow the CKMR literature, you are probably aware of one or several of these. So in 2018, Richard Hillary and colleagues used genome-wide SNPs and half-sibling CKMR to estimate abundance and mortality of white sharks around New Zealand and Australia. They also estimated juvenile survival using acoustic data. And then they combined their data from the acoustic tags with their CKMR model and alternative data sources on fecundity and young of year survival to estimate total population abundance. Recall that CKMR on its own can just estimate abundance of the adults. And so they ran their models over multiple population growth scenarios where they fixed the population growth rate. And this plot shows abundance of adults over the 5 different population growth rates. And these violin plots show abundance of the full population over 5 different population growth rates. And so I show this study to demonstrate that CKMR can be complemented by acoustic data, and that can increase the power of the analysis. Because acoustic data, in addition to allowing us to estimate survival, it can also help us understand movement dynamics if we want to build a spatially explicit model. Another neat application that came out recently was to spear-toothed sharks in Australia, where Toby Patterson and colleagues used genome-wide SNPs and half-sibling and full-sibling CKMR to estimate abundance and connectivity across rivers for this critically endangered species. Their model included parameters for adult survival, sex ratio, lucky litter effect, and effective female mates per male, though they focused their conclusions on abundance and connectivity. They also tried to estimate population growth rate, but they didn't have enough data, so they fixed it to 1. But in addition to demonstrating how CKMR can provide insight into demographic connectivity, this paper also showed that CKMR can generate results with a relatively quick turnaround time, and this can be very helpful for critically endangered species like spear-toothed sharks. However, it is important to note that when they started this project, the researchers already had a lot of the infrastructure in place from prior projects. So while it's true that CKMR can generate rapid results, getting the infrastructure in place to do that, particularly for the genetics and sampling, can be a heavy lift. And then the third example I wanted to highlight was to the thornback ray, where Trenkel et al. In 2022 gave us a great example of a study that was successful in spite of a number of hurdles because they had a thoughtful experimental design that included a number of checkpoints. So they originally planned to sample 7,000 thornback rays split evenly between adults and juveniles. But right when they were about to sample, their sampling plans were stifled for various reasons, and ultimately they had to abandon half-sibling pairs for abundance estimates. Fortunately, they had enough parent-offspring pairs that they were still able to use CKMR. But then, when they started analyzing the data, they found unexpected metapopulation structure. And so they went about their analysis then in 2 ways. One, where they split their data set into smaller data sets corresponding to the subpopulations, and then estimated abundance using CKMR separately for each subpopulation and summed the results. And then at the same time, they also just tried grouping all of the samples together and estimating abundance of the full metapopulation using the entire data set. And ultimately, they were able to obtain solid abundance estimates for 2013 and 2014 using both methods. And they were really quite similar, though their estimates for 2016 and 2017 were quite imprecise. And again, in the study, their estimates of population growth rate were imprecise, but overall, this was a successful application of CKMR. It's been used in a number of other applications that I don't have time to cover, but I wanted to list them here so that people have it in the recording. I didn't include the citations, but this should be, these terms in Google Scholar will get you the paper. And this is by no means an exhaustive list, and it does not include a number of neat simulation studies that have come out. But before we leave this section of the talk, I want to pause to discuss something that I've alluded to over the last few slides. And that is that population growth rate is possible, but it's challenging to estimate with CKMR. Some colleagues and I published a paper earlier this year where we used individual-based simulations to stress test simple CKMR models and then adapt them to account for factors that cause bias. And one of those factors was population trend. Now, recall from our half-sibling equation earlier that abundance is estimated in the birth year of the younger individual in each pairwise comparison. So in this case, we have the older individual of each comparison on the left and the younger individual on the right. So the N here refers to abundance in the birth year of the individuals on the right. If we have a full data set where we've sampled hundreds or thousands of samples, then we're likely to have hundreds of thousands or millions of pairwise comparisons, which means we almost certainly will have a number of different birth years represented in the data set. And that means if we want to use them in the same CKMR model, we need to connect the abundance estimates from each year using a population growth model. And this was the model that we used. It's a simple exponential growth model where abundance in year t is a function of abundance in year 0 times lambda. And when we think about our data set as a whole, the first year for which we can estimate abundance would be the birth year of the second oldest individual in the data set. And so that becomes our NT0. And it means that our adapted model looks something like this. So these violin plots show the relative bias that we observed after 500 distinct individual-based simulations with different population trends. And this relative bias refers to estimates of lambda. And we apply this to different sampling schemes where we assume that different age classes of our population were available to sample. So the green violin plots were scenarios where we assumed all ages were available to sample. The orange are when all juveniles were available to sample, and the purple is when we just targeted sampling to young of year over 4 different years. And whether the population was stable, increasing, or declining, our model produced unbiased estimates of lambda on average. However, when we introduced a severe population decline during the model time period, estimates were less accurate. In addition, as one might expect, if you look at the equation, estimates of population trend and abundance in year 0 were highly correlated. And when we dug into our individual results, we saw that in many cases, the model would overestimate lambda and underestimate abundance in year 0 or vice versa. But when they were multiplied together, when we derived abundance in year t, the resulting abundance estimates were unbiased. These results are not included here, but they're in the paper. And there's a lot of nuance to this. So if you're wondering how is that possible, like I can answer questions later. There's a lot of details I'm not explaining right now, but our big takeaway here was that if we're using an exponential growth model, it's wise to interpret estimates of population trend with caution, especially if older age classes are included in the data set, because then T0 is projected further into the past and transient dynamics can be obscured by long-term averaging of lambda. Of course, there are workarounds. We could include a quadratic term that allows population trend to rise and fall, or we also ran additional simulations in this project that showed that if data are sufficiently rich, if we have a long time series of genetic data, then we can process it using a kind of sliding window where we use 4 or 5 years of data at a time such that the long-term averaging effect is not so prominent. So there are potential ways to deal with this. And so now I'd like to step back again and then jump into the weeds of project design and consideration. I enjoyed this animation once I put it together, so that's why it shows up on every one of the roadmap slides. So we have one more instance of it coming up. So when we're trying to decide whether our system is appropriate for CKMR and whether it's worth exploring, there are a number of interrelated factors to consider. The first one, of course, is our sampling design. Sampling design includes how many samples do we need, what ages are available to us to sample, what locations will we sample, and how are we going to preserve tissue. And our sampling design informs and is informed by the type of covariate data that we need to collect. We pretty much always will need some kind of proxy for age. And if that proxy is taking length measurements, then that may constrain the number of samples that we can realistically collect. So this is a two-way arrow. And then the covariate data informs and is informed by the type of kin pairs that we want to model. And the type of kin pairs that we want to model depends on and informs the genotyping method that we should use. So if we're only modeling parent-offspring pairs or POPs, then we might be able to get away with microsatellites, which are kind of an older method of genotyping and assessing genetic variation. If we want to model half-sibling pairs or HSPs, then we need to use SNPs because there's a lot more of them. If we're collecting a lot of samples, then we almost certainly will need to develop a genetic panel. And if we're not able to preserve high-quality DNA, then we may not be able to use genome-wide SNPs, and that would mean that we cannot model half-sibling pairs. And so all of these different considerations are interrelated. And of course, we need to consider demographics and population dynamics as well. And so the main point that I want to make here is that there are a lot of things to consider when designing a CKMR project. And so it can help to, before we dive in, to step back and spend some time really digging into all of these interrelated factors and how it might realistically look. But let's assume that we've considered all of this and determined that we want to try to apply CKMR to our system. I now want to walk through the steps involved in planning and implementing a large-scale CKMR project. And as I'm laying this out, I'll highlight points like checkpoints in the workflow, as well as shortcuts that can be taken if the application is smaller. So the first step of a CKMR project would be to devise a sampling plan, And here we need to consider sampling infrastructure. How are we going to collect the samples? Will we collect samples invasively or non-invasively? And how will they be preserved? And remember, with CKMR, the birth years are more important than the sampling years. So when we're thinking about our sampling plan, we need to think in terms of birth years more so than the specific years we're sampling. The sampling years are most important just as they relate to our ability to age animals. And so the key point is that when we think about the temporal distribution of our samples, we should think in terms of the years that they were born, not necessarily in terms of the years that we capture them. An individual-based simulation can help us devise a sampling plan that is appropriate for our system. Once we have our sampling plan, then we can go out and collect a representative set of samples. And one of the big goals here is to dial in our sampling logistics. Oftentimes, we might have an amazing plan for our sampling that when we get out into the field, we see that it actually is not practical. And what representative means exactly will depend on the application. But in general, we want to capture as much of the genetic variation in our population as possible. Once we've collected our representative set of samples, then we can conduct lab work on those samples and sequence them. And a few of the goals here are to ensure that the DNA is of sufficient quality for our purposes and that we're getting the quantities that we need. It also helps to dial in lab protocols. These lab work protocols can be rather involved, and so having a kind of trial run with a small number of samples can really help us establish our workflow for when we have the bulk of our samples. And then at the end of this, we will generate genome sequencing data for our representative set of samples. And so these three first steps represent a kind of pilot project with two major checkpoints. The first is sampling feasible? Like, can we actually perform sampling the way we planned it? And the second is, are we getting the data that we need? Or is there something that's happening where our DNA is degrading? And if the answer to either of these questions is no, then we can circle back and revisit our sampling plan. And at this point, we hopefully haven't poured all of our resources into a CKMR project, but it gives us a chance to revise things. If we make it through steps 1, 2, and 3 and determine that we are getting what we need, then we can proceed with the next two steps in parallel. So when we generate genome sequencing data, we end up generating far more data than we need, and this drives the costs of sequencing up. We can mitigate those costs substantially by developing a genetic panel that targets future sequencing efforts to regions of the genome that are informative for our purposes. And when we do that, then for a given amount of sequencing effort, we generate far more usable data and far less unusable data with a targeted genetic panel than without. And this can provide substantial cost savings, especially if we're working with a lot of samples. And so we can use our subset of samples to develop a genetic panel. And this gives us a genetic panel that targets the regions, the SNPs that we've determined are informative for kinship. And this also gives us the chance to build up and refine our bioinformatics workflow. And at the same time that we're analyzing these data and developing a genetic panel, we can continue collecting samples. Once we've collected all of our samples and developed our panel, then we can conduct our lab work and sequencing for all of our samples using the panel that we've developed. And this can be done in batches as we go, or if we're concerned about batch effects, we can wait till all the sample collection is done and perform the lab work and sequencing all at once. And once we've generated sequence data, then we can get to work on genotyping and kinship assignment. And this is not trivial. In most cases, it will require access to a supercomputer cluster and a skilled bioinformatician. More and more papers are coming out showing that decisions that are made during the process of data filtering and genotyping can substantially bias downstream inference in ways that one might not think to check if they don't know what to look for. Kinship assignment is fairly straightforward when it comes to parent-offspring pairs, but much more involved with more distant relatives like half-siblings. So when it comes to genotyping and kinship assignment, it is wise to recruit an experienced bioinformatician. And then finally, once we are confident that we have assigned, that we know who's related to whom in our set of samples, we can fit our CKMR model to the data. Ideally, a model would have been developed and simulation tested at some point during this process. I showed one of the dozen figures from our paper earlier in this presentation, but one of the key outcomes of that paper was that we developed a CKMR model that is adapted to count for adapted to account for both population growth and intermittent breeding, even if the breeding schedule is unknown. This model is not nearly as complicated as it looks once you learn what the terms mean. And so this model that we present in the paper is a good starting point for applications to animals that skip years between breeding events. But in many cases, there will be additional factors that need to be accounted for. And that's kind of a general rule for the models that I'm showing during this presentation is they should be considered starting points, but not models that we should just go out and directly apply. There is not really a canned form of CKMR that can just be applied to all sorts of systems. So once we fit our CKMR model, then we can assess precision and determine if we've met our goals or not. And if not, we can just circle back to collecting more samples. And so this is the flowchart that I tend to follow with regards to planning and implementing a CKMR project. But of course, there may be shortcuts or alterations depending on infrastructure and expertise. If we have a small population, then we may not need to spend the time and effort developing a genetic panel. And so we can just skip right ahead. But how do we know if it's worth it to develop a genetic panel? So this chart from 2019 shows the costs for 4 different methods of genotyping. Microsatellites are the purple line, and that method has mostly been phased out, so I'd like to focus on the red, blue, and yellow lines. The red line corresponds to RAD sequencing, which is a method of reduced representation genome sequencing that generates all sorts of data, much of which we don't need for our purposes. And then blue represents rapture, which stands for rad-capture panels, which is one form of genetic panel. And then the yellow is a GT-seq panel, which is another type of panel. In this chart, it's the relative costs that I want to highlight more so than the absolute costs, which are always in flux. And if we look at the relative costs, we see that either genetic panel greatly reduces the cost of sequencing relative to rad-seq, and these cost savings become more pronounced the more samples that we include. And I should also mention for anyone who's been immersed in the CKMR literature that rad-seq is very similar to dart-seq, if you've read that before, and rad-capture or rapture is very similar to dart-cap. And so to determine if it's worth it to develop a genetic panel or not, it depends on our sample number, funding, and timing. But a good rule of thumb is that if we have more than 1,000 samples we plan to sequence, then it's probably worth it to develop a genetic panel. And if there's fewer than 500 samples, then it's probably unnecessary. And if you're somewhere between 500 and 1,000 samples, then you could go either way. But panel development does take time and resources. I have one colleague who is planning to do some genome sequencing for thousands of samples, and she's been working on a GT-seq panel now for over a year. But given the number of samples that she needs to generate sequence data for, ultimately it'll save money in the long run. And so when determining the number of samples that we need for a project, it really is kind of a circular way of thinking where we will likely want to apply CKMR in situations where we don't know population size, but we need to have a rough idea of population size to determine our sample size. So to give an idea of very rough sample sizes, these are the sample numbers on the y-axis needed to generate 100 half-sibling pairs, allowing for 15 false negatives, or 50 parent offspring pairs, and these correspond to a 10% and 15% coefficient of variation if we're fitting a very simple CKMR model, which in most cases we won't be. These are very rough numbers, and there are a lot of assumptions that go into these calculations, so they should not be used for anything other than obtaining a preliminary idea of a ballpark number of samples. Mark Brovington has called this, quote, the laugh test. But it can be helpful very broadly. Like, we may not know how big a population is that we want to apply CKMR to, but we might guess that there's over 1 million individuals in the population. And if we would guess that we have over 1 million individuals, but we only have access to about 100 samples per year, then this graph can inform us that CKMR is not likely to be feasible in this circumstance. But in most cases, this can give just a broad pass for if CKMR is worth exploring, but then we want to dig in more detail using like individual-based simulation to really dial in our sampling. So returning to our completed flowchart, I want to reiterate one more point. And that is that before we dive into any of this, it's a good idea to conduct a feasibility study to determine sample needs and definitely to ensure that we have the capacity to age animals, or at least have a plan for building that capacity in tandem with the rest of this. With regards to overall costs, the primary cost will be associated with personnel. It is a good idea to hire somebody to conduct a feasibility study first and kind of get all of these details in place and figure out what's worth exploring. And then we'll need personnel for sampling, lab work, bioinformatics and kinship, and population dynamics modeling. And these are not necessarily overlapping skill sets. Some people will be able to perform several of these steps, but in general, it's a good idea to consider these separate skill sets. For example, we don't want to assume that because someone can conduct lab work, that they can also do the bioinformatics and kinship. And similarly, if we know someone is computationally skilled at bioinformatics, we don't want to assume that they can do the population dynamics modeling. There are also, of course, costs related to supplies. With sampling, we will need sampling supplies and preservatives, but the biggest cost here, with marine species particularly, is likely to be boat time. So if there's an existing sampling program or survey that can be leveraged, that can help keep sampling costs down. And then we'll have costs associated with the lab work, including stringent quality control. And this includes kind of two categories of costs. There will be one cost for our sequencing of samples before we develop a genetic panel, and then we'll have another cost for per sample cost once we develop the genetic panel. And then we'll need computational resources for the bioinformatics and model fitting. So returning to our roadmap one last time, I'd like to spend the last bit of time here highlighting some ongoing applications of CKMR that I'm involved with. And I'm having a lot of fun with these projects, and I'm excited to give a brief overview to you all. So the first application, and the one that I'm most involved with right now, is for the third chapter of my dissertation. I'm applying CKMR to estimate abundance and mortality of white-spotted eagle rays in the Gulf of Mexico. They are a globally endangered species with largely unknown population trends. There is fine-scale population structure in the Gulf of Mexico has been suggested, but not confirmed. Something that I find particularly exciting about CKMR is that with a thoughtful experimental design, we can use the same data that we're using for CKMR to also delineate patterns of stock structure. But the reverse is not necessarily true, because the data requirements for CKMR tend to be a lot greater than for a stock structure analysis. We cannot assume that we can just tack on a CKMR study to an existing stock structure analysis. But with a little bit of thought, the opposite can often be true, and we can tack on a stock structure analysis to CKMR. We are fortunate to have a number of wonderful collaborators who have collected hundreds of high-quality genetic samples from 2010 to present. The fine-scale population structure that has been suggested in Mexico separates, or in the Gulf of Mexico, separates eagle rays kind of into two populations, one that is around Florida and the other that is over near Mexico. And so we have much greater sample sizes. These stars correspond to the locations we have samples from, and the numbers are the sample sizes. We have much greater sample sizes around Florida because that is the group that we plan to apply CKMR to. And previous studies have estimated effective population size around Florida to be between 2,000 and 3,000 individuals. Now, effective population size is a confusing term that is largely theoretical, and it has to do with the rate at which genetic drift occurs in a population. So it is not something that is a substitute for estimates of absolute abundance. But it can give us a rough idea of the order of magnitude of population size we're likely dealing with. And so given these estimates of effective population size and some individual-based simulations I ran, we determined that we likely have plenty of samples for CKMR. So last year, I spent a lot of time in the lab conducting restriction site-associated DNA sequencing on 742 samples. With 742 samples, we're kind of in that gray area between 500 and 1,000, and we decided that it was not worth the time and effort to develop a panel, so we just went straight through with it. And then recently I've been working on the bioinformatics and have identified a little over 9,000 SNPs for population structure analysis and a preliminary analysis of kinship. I'm still honing the kinship analysis a little bit. We don't necessarily want to use the same SNPs for both purposes, but we can get an idea of what we're looking at anyway. And so for the population structure analysis, it does seem like we have two clear populations. This is a PCA plot that takes the genetic variation and condenses it into a two-dimensional figure where samples that are similar tend to group together. And so here we see the samples from Belize, which is the star with the 7 next to it on the map, clearly separate out from the rest of our locations. When we remove Belize and the influence that it has on this plot, then we can start to see the samples from Mexico, the red dots in the plot on the right, also start to separate out a little bit from the samples around Florida. And so IRL and Sarasota are our 2 Florida locations. And so it does seem like we have clear population structure between the Gulf of Mexico and outside. And then we also seem to have some fine scale structuring between Mexico and Florida. And I've run a lot of different analyses, all of which support this trend, including analyses of FST, where the darker boxes show that we have more genetic differentiation between Belize and all the samples, and then a little bit between Mexico and the Florida samples, and really not much at all between the 2 Florida samples. In addition, it looks like we have about 25 relatives, 17 half-sibling pairs, and 8 parent-offspring pairs. But these numbers might change a little bit as I hone the kinship analyses. And so for our next steps, I need to formally assign kinship, refine and apply our CKMR model, and then ultimately the goal is to share our results with the IUCN to be included in their next Red List assessment. Another application that I'm fortunate to be involved with involves silky sharks in the Eastern Pacific Ocean, where stock assessments have been challenged by data paucity, but the Inter-American Tropical Tuna Commission is exploring the potential for CKMR to fill gaps. And so I've run a bunch of individual-based simulations of different population sizes with and without population structure and compared the number of kin pairs for different sampling schemes. And I'm not going to break down this whole plot except to highlight that all of the different plots show different scenarios of what the populations might be doing. And the highlighted cells between blue and red show the number of kin pairs that we're identifying. And I tried to highlight cells that would generate a number of kin pairs that are likely to give us the results we want without going overboard. And so now we're working on getting sampling supplies into the hands of folks who can actually collect the samples. I've also had a number of conversations with people about potential applications coming up in the future that I'm excited about. And these include scalloped hammerheads, dolphins, and brown bears and wolverines. So there is a lot of interest in CKMR, and that interest is just continuing to grow. And so if we return one last time to our handy dandy flowchart, the Eagle Ray project is down at step 6, where I'm finishing up bioinformatics and kinship assignment, and soon we'll be transitioning to step 7. The Silky Shark project is a step 1, and we're about to transition to step 2. And then the potential applications are back at step 0, which is seek funding. So over the last 45 or 50 minutes, we have covered a lot of ground. And so I want to leave you with a few of the major takeaways here. And the first takeaway is that CKMR is a highly adaptable framework, and it does have a lot of promise for allowing or improving estimates of abundance, mortality, and other parameters. But it is very important to spend time designing the project and to make sure that we have the appropriate expertise to carry out the project. I do want to emphasize that lab work, genotyping, and kinship assignment require time and expertise. And these are not necessarily tasks that can just be given to any genetics lab, but there should be conversations beforehand if the genetics are being outsourced to make sure that the appropriate expertise is available. And in large-scale applications of CKMR, development of a genetic panel will likely be required to keep costs reasonable. And an initial feasibility study can help ensure that resources are not wasted, and simulation can help. I have a custom individual-based simulation code, which is all written in R, that I've found very helpful. It takes as input various vital rates and sampling designs, and then it simulates a population forward in time and tallies the number of parent offspring, half sibling, and full sibling pairs. At some point, I'd love to get this package up nicely. I'd love to package this up nicely and get it on CRAN. But until then, I'm happy to share it with anybody who's interested. My contact information is on the next slide, so just reach out if you're interested. This code is not in a place where it's ready to just plug and play, so you have to be ready to dig in a little bit, but I'm happy to talk that through. And then lastly, it is helpful to include checkpoints in a large-scale CKMR project so that things can be adapted and optimized as needed. And so thank you all for taking the time to listen to me, and thank you, Lisa and Bai, and the organizers for your time.
Thank you, John. I'm going to go ahead and stop your screen. You can go ahead and put your webcam on. Hi, everybody. We have about 6 minutes for questions, and I hope you have them because this was a fabulous presentation. We did get actually an example of let me send this across the screen. This came in while you were talking about 17 minutes ago, an example of using a form of stock synthesis. And I'm going to actually put the link to that article here in the chat. This is a article that comes from Fisheries Research called Including Close Kin Mark-Recapture Data and Statistical Catch at Age Stock Assessments and Management Strategies.
Cool. That's the paper by Andre Punt, right?
Yes.
Yeah, that's a fantastic paper.
All right, so I'm starting to see some questions coming in. Let me go ahead and approve them. And let's see. The second question asks, I noticed adult survival is in the population estimation equation. How sensitive are results to this parameter, which is notoriously difficult to estimate, both from data and from models?
How sensitive are results? So in terms of, I think one of the keys for survival estimation is having enough cohorts. This is one of the things with our paper, like we had that sampling scheme where we targeted sampling to young of year individuals for 4 years, and that seemed to work for abundance estimates, but it did struggle with estimates of survival. But overall, when we had more cohorts in the data set, we found that survival was estimated pretty solidly within the CKMR model. Yeah, so it seems like results are not, my experience has been they're not super sensitive to it, but I didn't talk about a lot of the details involved in survival estimation because it can get a little bit weird. If you have too few cohorts, then survival and abundance become very highly correlated and neither becomes really reliable. But we had overall, and these results are in our paper as well, we found that survival seemed to be estimated pretty reliably if you had enough cohorts.
Thank you. This next question asks anonymously, great talk, well laid out. What sample size would you say is necessary for kinship identification to be successful? I can imagine if the population is large, it might be hard to find any relatives.
Yeah, well, that is exactly why I included that slide that showed the sample size by population size, because there's not really one sample size that's necessary for kinship identification to be successful. It all depends on the population size. The larger the population, the more samples we need to identify kin, and this also will depend on how the population behaves too. And so this is exactly why this question that you're asking kind of gets at the heart of designing a CKMR experiment, and it's why individual-based simulation and a feasibility study are really important to consider upfront.
Thank you. Oops. We get this next question out here. This next question asks, you mentioned a few times the potential limitation of collecting age data. Has anyone considered using DNA? Ooh, this is a challenge one. Methylation as a proxy for age. It would add cost on the lab work sequencing side, but may reduce sampling effort considerably.
Yes, I am really happy somebody asked this question. Yeah, there has been a lot of talk about using DNA methylation as a proxy for age, because if you can do that, then you can also conceivably use the same tissue samples that you're using for CKMR and potentially stock structure also to age the animals. So there is definitely interest in this. One of the one of the tricks about it is that you have to get a precise methylation clock. You often need to develop it for the specific species that you're applying it to, and that requires a fair bit of upfront work and validation as well, where you look at methylation, you identify sites that are informative for age, but you need to then have samples of known ages that you can compare it to. And so there is, I'm actually involved in a project right now where we are designing like CKMR sampling, where we're also talking about DNA methylation. Um, and so, so people are definitely talking about this, but in studies where we want to use DNA methylation, we need to keep in mind that there is a lot of calibration up front that needs to happen to do this reliably.
Excellent. Thank you so much, John. I think that was our last question for today. Oh, actually, give me
I
one
see one
moment.
more.
Yes, I do. It just came in. I'm just under the gun. This one last question asks, give me one moment, from Matthew. It seems like the CKMR is reliant on 100% correct assignment of relatedness. Have there been any studies looking at the sensitivities of these models to inaccuracy in assigning relatedness?
Yes. So I highlighted just briefly two of them throughout the talk. One of them was the paper that some colleagues and I published earlier this year, where we misassigned ages and then did not account for that in the model at all. And we found that, that does bias our abundance estimates. But then another researcher, Felix Pietersma, a couple months ago, put out a fantastic paper that showed that if you have an age length curve, even if it's not particularly precise, you can build that into your model so that if there's some uncertainty around aging, the model will account for that. And so yes, so there. If we account for imprecision in aging, then then we can build a model that is robust to that.
Excellent. We are getting some more questions, and I'm going to go a little long today just because these are great questions. One question came in the chat, so I'm just going to read it out loud to you. Jim asks, how much of an impact does aging uncertainty create? Do animals that have long lifespans create more issues than shorter life species when many samples will come from adults?
Cool. Yes, so I will answer that in a moment, but I realized the last question I just answered for inaccurate aging, and someone just informed me that the question was about incorrect relatedness. And I am not aware of any study that has formally looked at this, but there is a lot of discussion about incorrect relatedness and challenges with false positives in some of the papers that Mark Brovington and company have put out. And so when this becomes especially important, like for half siblings, because you can find false positives can be a problem with half siblings, but there is, of course, that trade-off between type 1 and type 2 error. And so Mark Brovington and company, they've designed a package that assigns relatedness that's robust to false negatives. So you can kind of lean a little bit more towards false negatives to decrease your chances of false positives, and assigning relatedness in the model can then account for that. So I'm not aware of any study that has formally tested that, but we do, especially with half siblings, have to be real mindful of potential false positives, because sometimes we're collecting like thousands of samples and we've got just a handful of related pairs. And in that case, having one false positive or two can add a greater bias than we might hope for. And then the question back to aging uncertainty.
Yeah, I just have it right there.
Yeah, great, thank you. It can, so do animals have? Okay, I'm going to kind of speculate on this. I would need to think a little bit more to really answer this in detail. But I think in general, if animals have long lifespans, then in many cases, aging is harder the older animals get. And so you would have more uncertainty with aging if you're sampling older individuals. But then there's also the issue that abundance is estimated in the birth year of younger individuals in each comparison. So if your data set tends to comprise mostly older individuals, then you're going to project abundance back into the past quite a lot. And so I imagine that these two things would interact with one another, but I would have to, I think I'd have to think about it think about it a little bit more.
Excellent. Well, that's going to be our last question today. John, you did a fantastic presentation today, and I really appreciate it. Audience, I want to thank you for your great questions and for staying a little bit long, but I think it was worth it. John, we appreciate you here at NOAA, and Bai, thank you again for bringing John to NOAA and today's presentation. Join us again next month, September 5th, which is the first Thursday of the month at 3 p.m. Eastern Standard Time for the next National Stock Assessment Science Seminar. And if you do have any questions for John, we did put the email address in the chat, but just to make sure it's jswenson@umass.edu. Thank you so much, everyone. Have a wonderful rest of the day. Bye-bye.